हेलो गाइस, आई एम शुभम फ्रॉम शो ट्यूटोरियल्स। इस वीडियो से हम रेप्लिकेशन PYQs स्टार्ट करेंगे। बेसिकली वी विल डिस्कस 10 क्वेश्चंस पर वीडियो। So this is the part 1 of this series. Okay, we will be covering all the molecular biology PYQs of the CSIR net, right? So before we start, subscribe to our channel and press the bell icon to get notified whenever new videos out. This is absolutely free from your side, but this will help us grow. First question for today: copying errors occurring during replication are corrected by proofreading activity of DNA polymerases that recognize incorrect bases. Okay, we know proofreading activity जो mispairing होती है that correction takes place from, right? Now we know that जो synthesis है that takes place from 5 prime to 3 prime. Okay, so 3 prime end पर nucleotides add होते हैं. So if proofreading activity has to be occurred, then 3 prime to 5 prime होना चाहिए, right? जो removal of the incorrect bases that should occur from 3 prime to 5 prime. तो जो options हैं, first option is at the 5 prime end, so this will be wrong. Second, at the 3 prime end of the growing chain, इतना correct है. And remove them by 5 prime to 3 prime exonuclease activity, no, so wrong again. Why? We saw 3 prime end पर होना चाहिए, remove them by 3 prime to 5 prime exonuclease. Option 3, add the 3 prime end to the growing chain and remove them by 3 prime to 5 prime exonuclease activity. Yes, so this is correct. Add the 5 prime end, again wrong. Okay, ये पूरा डिटेल में हमने डिस्कस किया है, right? In the molecular biology playlist you will find the videos. Second question, you have labeled DNA in a bacterium by flowing cell in a medium containing either 14 nitrogen or the heavier isotope. Okay, two different isotopes are used. So N14 here, that is lower, and N15 is heavier. Furthermore, you have isolated pure DNA from this organism. DNA को isolate किया and is subjected to CSIR density gradient centrifugation, leading to separation of light and heavy forms of DNA to different locations in the centrifuge tube. ये light and heavy forms का separation हुआ. In the next experiment, bacteria were regrown first in medium containing N15 so that all DNA made by the cells will be in heavy form, right? N15 में grow किया तो heavy form of DNA उसमें होगा. Then these cells were transferred to medium containing only N14. Again transfer किया light nitrogen में and allowed the cells to divide for one generation. DNAs were extracted and centrifuged again as above in the CSIR gradient. Now hybrid DNA band was observed at position located between and equidistant from the N14 and N15 bands. Based on the observation, which one is the of the following conclusion is correct? Okay, replication of DNA is conservative, semi-conservative, dispersive or circle mode. See, very easy. I will draw your N14, N15. Okay, this is a, we can say, a banding. So in the question it is mentioned that hybrid DNA band. Okay, मतलब N14 एंड N15 के बीच में यहाँ पे हमें बैंड मिला, विच इज इक्वल डिस्टेंस फ्रॉम N15 एंड N14. इक्वल डिस्टेंस है फ्रॉम हियर एज वेल एज हियर, इक्वल डिस्टेंस. बेसिकली दिस इज सेमी कंजर्वेटिव एंड बिकॉज यहाँ पे N14 भी प्रेजेंट है, N15 भी प्रेजेंट है. बोथ द isotopes are present in equal amount, right? So this is a semi conservative mode of replication, right? Although ribonucleoside triphosphates, that is RNTPs, are present tenfold, approximately tenfold higher concentration present than deoxyribonucleoside triphosphates. Means जो RNTPs है, these are more in number than DNTPs, but they are incorporated into DNA at a rate that is more than 10,000-fold lower than DNTPs. This is because मतलब incorporation फिर भी जो RNTPs का है lower है than this one. Why so? Let's check the option. DNA polymerase cannot discriminate. Wrong. We know that DNA polymerase discriminate कर सकता है between these two. Okay, DNA polymerase is a specialized enzyme, right? So it can discriminate. So again, DNA polymerase cannot discriminate. Wrong. Next, DNA polymerase efficiently discriminates between RNTPs and DNTPs. See, from statement also you can understand. Okay, अच्छे से रीड करना है, right? फर्स्ट एंड सेकंड स्टेटमेंट में कैन नॉट डिस्क्रिमिनेट दिया है, रॉन्ग है. यहाँ पे दिया है efficiently discriminates because its nucleotide binding pocket cannot accommodate a 2' OH on the incoming nucleotide. See, very accurate statement. तो न्यूक्लियोटाइड बाइंडिंग पॉकेट होता है, उसमें 2' OH accommodate नहीं हो सकता. So this is very correct, right? The fourth statement again says DNA polymerase cannot discriminate. It's again wrong. Okay? हमने देखा है DNA polymerase का स्ट्रक्चर, then nucleotide binding pocket क्या है, how it differentiates between RNTPs, DNTPs, right? All we have seen. Next question. In eukaryotic replication, helicase loading occurs at all replicators during G0 phase, G1 phase, S phase, G2 phase. We know that helicase loading G1 phase में होती है. Helicase activation, remember this thing, very important. Helicase activation S phase and loading in G1 phase. Next, in all organisms, it is critical that replication initiation be tightly controlled to ensure that chromosome number and cell number remain appropriately balanced. Given below are several statements regarding regulation of replication in E. coli. Okay? We know that there is methylation and hemimethylation states. Okay, so let's take the statement. What we have to do, which of the above statements are not true? A statement, hemimethylation and sequestration of oric, that is origin of replication, by protein called SEC prevents initiation of replication. Yes, so hemimethylated state है, that does not occur initiation, right? So that is prevents initiation of replication. So this is correct. Availability of DNA A protein is an important requirement for initiation of replication. Yes, DNA A protein initiator है. A and B are correct statement. Let's see C. The ratio of ADP-ATP is important as high level of ADP is required for initiation of replication. Wrong. C. High level of ADP नहीं. So the correct statement should be instead of ADP, it should be ATP. ATP requirement is important for replication. Right? No ADP. So thus it is wrong. Recruitment of HDA protein by sliding clamp inhibits ATP hydrolysis required for initiation of replication. Inhibits नहीं, प्रमोट करता है. ATP hydrolysis को inhibit नहीं, प्रमोट करता है. So D is wrong. So what is not true? C and D. Option number 3. It takes 40 minutes for a typical E. coli cell to completely replicate its chromosome. Simultaneous to the ongoing replication, 20 minutes of fresh round of replication is completed before the cell divide. What would be the generation time of the E. coli growing at 37 degree in complex medium? यहाँ पे दिया है 40 minutes required for complete replication, right? But second statement में दिया है ongoing replication, मतलब ongoing replication जब चालू है तभी 20 minutes of fresh round of replication is completed before the cell divide, right? मतलब 20 minutes ongoing replication, एक replication जब चल रहा है तो दूसरा वाला भी चल रहा है ऐसा. So these are overlapping basically. So 20 minutes of fresh round of replication is completed before the cell divides. So the generation time will be 20 minutes. Carefully read the question. 40 minutes for complete division, but 20 minutes में fresh round of replication होता है simultaneously, right? So the generation time will be 20 minutes. Again, next same type of question. E. coli takes 40 minutes to duplicate the genome using a bidirectional mode of replication. Okay, bidirectional takes 40 minutes. If E. coli were to use unidirectional mode to synthesize a full copy of DNA complementary to just one of the strands of the genome, it would take. See, bidirectional को 40 minutes. Then if single करना है, then it will be double, means 40 plus 40 is equal to 80. Okay, so option number 2 will be the answer. And unidirectional मतलब पूरा करना है. This is a circular chromosome. If bidirectional has to occur, this take 40 minutes. अगर सिंगली पूरा करना है, ये 40 हाफ का 40 plus this 40 will be 80, right? Half. Last question for today. In E. coli grown under nutrient-rich condition, replication of entire genome takes about 40 minutes. Same type of question, yet it can divide every 20 minutes. This is so because, why this happens? While E. coli divides every 20 minutes, equal transfer of genetic material occurs only in the alternate rounds of cell division. See, ऐसे स्टेटमेंट्स में हमें carefully read करना है and most accurate statement you have to identify. Okay? And इससे ही आपकी जो problem solving skill है that will increase, right? So you have to be aware of statements you are reading. सेकेंड यहाँ पे ओनली है, तो दिस इज एक्सट्रीम टर्म, ओनली ऐसा कुछ नहीं होता, राइट? मीन्स वेरी स्पेसिफिक क्वेश्चंस आर देयर which can be solved by only thing. Next, a second round of genome replication begins before the completion of first round. जो प्रीवियस क्वेश्चन में था साइमल्टेनियस रेप्लिकेशन होता है, second replication begins before the completion of first replication, and by the time cell is ready to divide, 2 copies of the genome are available. So this statement seems to be more accurate. Okay. Then also we will check the third, genome replication and cell division are not coordinated with each other. See, this is so wrong. Cell division and replication में coordination नहीं है, ऐसा नहीं होता. Okay. जो cell division का second phase है, S phase, उसमें replication होता है, right? And these are coordinated. Next statement, during cell division, only one of the strands of the genome whose synthesis can be achieved in 20 minutes is transferred to daughter cell. Only one strand. Okay. Wrong. These are not accurate statements as well as one. Okay. Only in the alternate round. Not coordinated. Then only one strand. These are extreme statements. They are not true. Second, when you read carefully, you can understand ये बहुत accurate statement, right? So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section. Like this video, share this video with your friends, subscribe to the channel, and stay tuned for the upcoming videos. Thank you.